No, you'll, yeah.
Yeah, I can go to screen, but no, when
Very
you're finished,
good. Well, I think we are live now.
Okay.
Okay, super, super. Greetings to you all. Good afternoon, good evening, or good morning, depending where you are. Welcome to this webinar. And today we have a special guest from far away, from Lincoln University of New Zealand. And welcome, Professor Leo Condron.
Hello.
We thank you very much, and we appreciate very much your availability at this time of the day. The hour there is past midnight. It's almost 1 o'clock.
Yep.
You are there fresh and awake. Webinar. Thank you very much.
I'm not so
we
sure.
Appreciate that very, very much.
No worries. Okay.
Okay. So let me just give you a brief overview of a short biography of Professor Leo Condron, if you may. Leo Condron has been an academic at Lincoln University since 1992 and was appointed to a personal chair in biochemistry in 2006. His research has focused on investigating the dynamics of organic carbon and major nutrients in natural and managed ecosystems, with an emphasis on the nature and bioavailability of organic and mineral forms of nutrients in the soil-plant system in relation to soil management and land use. Project areas of Professor Leo Condron include organic matter and nutrient dynamics in grassland and forest soils, soil chronosequence dynamics, rhizosphere processes and nutrient acquisition, relationships between soil microbial diversity and function, and the availability and mobility of phosphorus in terrestrial environments. Today, he will lecture to us about the role of biology and organic phosphorus in determining the dynamics and bioavailability of phosphorus in soil-plant systems. And we're looking forward to your talk. And thank you again for accepting to give this talk late at very hour from New Zealand.
Okay.
So, cool. Without any further ado, I will give you the floor.
Okay.
And you're there.
Thank you, Mohammed. I'll just get things started here. Yeah, here we go. So, thank you for those kind words. And it is a pleasure to talk to whoever I'm talking to. Yep. I've never done a webinar before, so please excuse my slight hesitancy sometimes. Anyway, so as you explained, I just want to talk a little bit about the role of biology and particularly organic phosphorus in phosphorus cycling or dynamics in soil plant systems. And so, I mean, you know, this is one of many representations of the phosphorus cycle. And the point I just wanted to make about this is that unlike nitrogen is almost in soils is almost exclusively the bioavailability and dynamics of nitrogen is almost exclusively controlled by biological and biochemical processes. Sulfur is similar, but in the case of phosphorus, we have a combination of mineral systems here, chemical reactions, if you like, and biological and biochemical reactions. And it's that interaction between the two that that determines the dynamics and bioavailability, and indeed the mobility of phosphorus in these systems, soil plant systems. And just to put some numbers on it, and I think it is important to put some numbers. This is an example from Australia of a grazed pasture system in kilograms of phosphorus per hectare per year. And it just gives you the oh, sorry, I've lost my screen here. Oh, there we go. It gives you an idea of if you look at this system, you have variable inputs of fertilizer, for example, 10 to 30 kilograms of phosphorus, which almost matches the uptake by the plant. But you'll notice that we've got residues here on the left-hand side, and basically most of the phosphorus that's taken up by the plant is actually returned to the soil in plant or animal residues. Very little is lost in outputs, and either if we use a graze system or we use indeed a cropping system. So most of it goes back into the soil. And if you look at the soil, we can consider it as three components, really. You've got the what I call the biomass phosphorus, which is the phosphorus that's inside living organisms, which might constitute 10 to 30 kilograms of phosphorus per hectare at any one time. And you've got the inorganic phosphorus, 100 to 200, and the organic phosphorus, 100 to 200. The important point is that in many soils, approximately half of the phosphorus is present as inorganic phosphorus, and about half is present as organic phosphorus. So I'm going to concentrate. It is very difficult. It's almost impossible to separate them, and that's one of the challenges of working with phosphorus, is that these chemical processes are intertwined with the biological and biochemical processes. But what I'm going to focus on is the organic side of things. So if we look at that phosphorus cycle, what I'm doing is I'm just going to concentrate on the organic side of the system for the moment. So I'll just blow that up a little bit so you can see it. This is quite a complicated diagram, but it basically, again, it looks at the different forms of organic phosphorus, and I'll explain a little bit more about what I mean by organic phosphorus in a minute. But there are basically, if you like, three different forms. There's most of the phosphorus that's contained within the microbial biomass, and microbes, bacteria, fungi, and archaea make up 95% of the microbial biomass in soils. So we're talking about mainly microbial biomass here. Is organic, and then in the soil itself, you've got the non-living organic phosphorus, and you can split that into two broad pools. There's an active pool, which is some maybe call a labile pool, some of the terminology, and then there's a stabilized pool, which we might call the more recalcitrant organic phosphorus. And a point I'll come back to a little bit later is that the release of phosphorus from organic forms, these active or stabilized forms, is driven mainly by the actions of phosphatase enzymes, and these enzymes are produced mainly by microbes in the soil, but also by plant roots as well, and things like mycorrhizae and also rhizobia as well. So this is what I'm going to concentrate on a little in the next few slides as well. And coming back to that stabilized versus the two different forms of phosphorus, the active and the stabilized, we can just to simplify things, I think what we can talk about is that the active material is what's known as mainly diester phosphorus. And what we mean by that, and this is an example of DNA, is that the phosphorus is actually connected to carbon through oxygen and through carbon by two links. So it's known as a diester, and monoester, the stabilized, is actually phosphorus that's connected to carbon, in this case carbon in a ring, by one link. And you might think that phosphorus that's connected at 2 points to carbon might be more stable than connected to one, but one of the main factors, and I'll come back to this a little bit later, that makes things like inositol hexaphosphate very stable in soil is that it's actually absorbed onto soil minerals in a very similar manner to inorganic phosphorus. So that's what stabilizes this inositol hexaphosphate. And we can see that the stability of that if you actually look at the total organic phosphorus in some bacteria, E. coli, representative fungi, and two plant species. And here you can see that nucleic acids or diesters and phospholipids are another form of diester. They make up roughly about 80% of the organic phosphorus. That's the organic phosphorus that's in those organisms. 80% of it is diesters, and somewhere between, say, roughly 10 to 25% is monoesters. When we look at the soil, and thus far we've only been able to characterize just over a half of the organic phosphorus in most soils, but the vast majority of that, what we've been able to characterize in soil, is actually monoesters. And what that tells us is that diesters, so the organic phosphorus that's coming into soil is coming in bacterial residues, fungal residues, plant residues, animal manure, etc. is coming in. It's mainly diester phosphorus, which is broken down in the soil, and there's very, very small amount residual, whereas monoesters are actually quite stable in the soil environment. So I'm going to explain that a little bit more by using an example. And just to come back to the point of how organic phosphorus makes a contribution to plant phosphorus requirements is mainly through this mechanism, which is basically that a plant has, if there is low levels of low supply of phosphorus in the soil solution, then basically the plant roots and and its associated microorganisms in the rhizosphere produce enzymes, extracellular enzymes, phosphatase enzymes, which are designed to release phosphorus from organic sources and thereby increase the supply of inorganic phosphorus to the plant. Plants and microbes can only take up inorganic phosphorus, so they can't take up organic phosphorus. So it has to be broken down first by these extracellular enzymes. The inorganic phosphorus increases, and when that increases, the plant can then take up the inorganic phosphorus, and it turns off the enzyme production. So it's known as a negative feedback, because enzymes are very expensive in terms of the metabolically and nutritionally, actually very expensive for microbes and plants to produce. So they're only produced when they're actually required. So that's how the enzymes work. And again, I'll come back to that a little bit, refine that story a little bit towards the end. One system for looking at the role of organic phosphorus and the role of microbes in phosphorus nutrition of plants, essentially, is to actually look at natural systems. And if we look at the sort of 5 soil forming factors, which are parent material, topography, climate, and organisms, and the 5th one is time. So if we can get an environment where we can hold these reasonably constant, not necessarily rigid, and vary over time, then we can start to look at how, particularly an element like phosphorus, which if you look at a natural system, the phosphorus that's in the parent material is basically that is essentially all the phosphorus that you've got, that the system has. There is some minor input of phosphorus in the form of dust in the environment, and we know that dust is actually very important. But so we do have some small inputs, but for the most part, phosphorus, unlike nitrogen, which can be fixed from the atmosphere, you're limited to the phosphorus in the parent material. So if you can look at a sequence of soils over time, you can get some idea of the phosphorus dynamics. And this is a well-established phenomenon in terms of chronosequences. And there are many examples of this. I'll just highlight 2, and I'm going to talk about one in detail. The top 3 here is basically glacial deposits. I'll explain that, and glacial moraines that are left behind by glaciers, where you can actually look at the development of ecosystems from basically parent material to mature forest. The second example down here is actually a sand dune system in coastal Eastern Australia and Queensland, where basically you've got sand being deposited continuously on the coast. So what happens is when it gets deposited on the coast, it then gets becomes a forest. It becomes invaded, if you like, by trees, by plants, and over the course of time, that coast keeps growing out. So the further you go inland, the older the forest gets. So you get a sequence of forest development. But just to show you what happens in these ecosystem development things, this is a bit of a complicated diagram with a lot of things on it. But basically, when you start off with just a parent material, and this applies to volcanic materials as well, ash and lava as well. You have a couple of things you have. One is that you virtually have no nitrogen and you have no carbon. Where's my carbon? There you are, down there. But the one thing you do have is phosphorus. Phosphorus is in the parent material in the form of apatite, and there's a reasonable supply of available phosphorus. So what happens is the first plants, or the first well, actually probably algae or whatever that invade, that colonize that parent material, is actually nitrogen fixers. So they fix nitrogen from the atmosphere, and through that process, and through the process of legumes, we start to get a biomass development. We start to get a scrub and then a forest develop. And what happens is then that we accrue nitrogen from the atmosphere. The soil carbon increases as the plants turn over. The microbial biomass increases. Net primary production and above ground biomass. But what happens with time is that because there's very little of essentially no phosphorus coming into these systems, the available phosphorus starts to decline. And I'll explain that in more detail in a moment. And what happens eventually is that these systems, initially they're nitrogen deficient, and then they go through a phase where there's adequate supplies of nitrogen and phosphorus. But in time, they become phosphorus limited and phosphorus deficient. And so what happens is that over the course of time, these systems start to actually decline. And this is what's called retrogression. They start to decline. The primary production starts to decline. The carbon, the biomass, etc. So these are ideal systems to actually look at phosphorus dynamics in. I'm just going to describe one to you. We'll work on a number of them here in New Zealand and also in Australia. But this is one which is quite unusual in that it's a series of glaciers on the west coast of the South Island, which is actually almost at sea level. So you can, they're very accessible. A lot of glaciers in the world are at high elevation. These are, well, pre-COVID, they were a big tourist attraction for overseas tourists to come and look at these glaciers because they're very, very accessible, very easy to get to. But if you've never seen a glacier before, this might disappoint people because basically this glacier, the glacier on the right hand side here, the glacier is actually just around the corner, just around the back of the valley here. But what happens is this ice basically mass moves up and down the valley over the course of thousands of years, and as it moves down the valley, it basically grinds up rock material. So when it melts, it leaves behind this ground up, well, not ground up rock. There are particles of rock in it as well. And over the course of time, these deposits have been left at different times throughout the valley. And if you can identify these different surfaces, then you can actually have a series of different aged, in this case, forest environments. And let me try and explain that a little bit better. This is a graph. So basically, if I go back to this previous picture here, on the left-hand side, that's the front of the glacier. So this glacier is retreating. It's actually retreating at the moment. like most glaciers around the world. But it's retreating, and when it retreats, it leaves this material behind. So that's essentially, if you like, the front of the glacier here, where it says 1972 on the left-hand side there. But if we go down the valley, we can actually identify. We can use tree rings actually to identify trees up to 600-700 years old, and we can use carbon dating to actually find surfaces that are older than that. So if you look on the picture on the right-hand side, is actually an expansion of this. So this is the front of the glacier here. If you go all the way down that valley, you'll find surfaces all the way and way out here, which is getting out towards the coast. There is a site. So at the glacier front, we're talking about time zero, and way out here, we're talking about 120,000 years. So you've actually got 120,000 years of soil development here with very little phosphorus input in this system. This system was first described in the 1960s, and it's fair to say it's fairly well known around the world. There's still a lot of studies going on, different aspects of being studied on this system. The soils, they start off just as glacial till, and you can see it's not all ground up rock. There are rocks there too. And basically what happens is, as it develops over the course of 120,000 years, you basically get a forest developing, organic matter, organic layer, you get podsilization occurring. So it's a fairly well-defined sequence. And if we look at some numbers here, and you'll see that we don't have even numbers. We have ages from 5 years up to 120,000 years old. But it gives the idea that you see the carbon, there's a little bit of inorganic carbon in the system, but most of very little organic carbon and nitrogen at the beginning, but there is adequate levels of phosphorus, 800 parts per million. That's quite a lot of phosphorus. But what happens is that over time, the forest starts to develop, the nitrogen starts to be fixed, so nitrogen goes up, carbon increases, and you'll notice that phosphorus starts to decrease. And the reason why phosphorus in the soil decreases is partly because of biomass uptake, but also mainly because of loss in drainage. These soils have been estimated are losing about the rainfall at Franz Josef is about 3 meters per year. So it's losing about 1 kilogram of phosphorus per hectare per year. So we're getting a loss of that's your main loss of phosphorus. So what happens is the forest develops and the biomass, the ecosystem develops, and it's again, first of all, nitrogen deficient, and then in this area, somewhere between 500 and 5,000 years old, around about say 500 years, this one here, it's basically there's adequate levels of phosphorus, there's adequate levels of nitrogen, so that system is actually at what we call its peak. It's basically at its peak biomass. What happens after that is somewhere between 500 and 5,000 years, the system tips into retrogression. In other words, phosphorus starts to become limiting. And as it continues, the phosphorus continues to be lost by drainage. So by the time we get to 120,000 years, we have a system where we've lost 90% of the phosphorus from the soil has gone. Some of that will be in the biomass, but only a small proportion, only a small amount. And you'll see that the carbon has peaked and then gone back down, and nitrogen similarly has gone back down. So this is the system that we have here. So if we look at it in terms of phosphorus, and this was data that was published way back in 1970 in the 1970s, when they looked at phosphorus in the profile, you can see that there are basically 2 phases in this system. The first phase is when the ecosystem, the forest is developing, and you've got at the beginning 100% of the phosphorus is apatite phosphorus. It's not water soluble, but it's very readily available to plants and to microbes, and basically it starts to dissolve, and you also get weathering. You start to get iron and aluminium oxides forming, so you start to get these secondary mineral forms. These are inorganic forms of phosphorus, mainly associated with iron and aluminium, and initially these are quite available, but they become less available with time. And we also start to get some organic forms of organic phosphorus appearing as well as the biomass develops. But at some point, when this P limitation starts to come in, we're losing total phosphorus because of drainage loss. Then what happens is the system changes to being phosphorus limited. And that's a very important change. It's not a dramatic change like it's portrayed here. It's a gradual change. But the way I like to think of it is that this first part of the system here is mainly driven by chemistry. The dynamics of phosphorus and its bioavailability is mainly related to chemistry. And there's some biology involved in it, obviously. But when we get to this latter part here, these secondary mineral P forms are very, very stable, right? So the continued productivity of that system and survival of that system is actually dependent on organic forms of phosphorus. So we have a chemical system, then we have an organic or a biologically dominated system. And that again makes it a very interesting system to look at, particularly what happens to this organic forms of phosphorus. And just coming back to my diagram again, the thing I showed you before about these active or diester phosphorus, that's the phosphorus that's coming in, the organic phosphorus coming into the system. Then we have the stabilized ones, which is the monoester forms of phosphorus. But what we can do is we can actually, and what we've done is we've actually looked at the nature of organic phosphorus as we go along this system to try and get some idea of how the relative the nature of phosphorus in these systems changes. And what we expect to see is that as the system gets older, if I can flip back to this one here, as it gets older, we expect to see possibly less and less of the labile organic phosphorus and more that these stabilized forms. In other words, it mimics what happens with the mineral phosphorus. We get more and more stable organic phosphorus as the ecosystem gets older. Well, in a classic case of a hypothesis being wrong, what we found was exactly the opposite. And let me deal with the monoesters first. In terms of the monoesters, in two examples here, what we found, we expected to find that these would increase with time. So this is time along the bottom here. What we found was that in point of fact, they decreased, and especially towards the older part of the sequence, they dramatically went down, it decreased. And this was a little bit puzzling, but we think that the reason for that, if we go back, if you look at some other characteristics of the sequence, and one of the things is that the pH decreases from nearly 7 down to 4. And if you look at amorphous iron and aluminium, they'll initially increase, but then particularly for aluminium, it decreases with time, and particularly towards the older part of the system. And I mentioned before that these inositol phosphates are stabilized by adsorption onto mineral surfaces. So amorphous mineral surfaces. So as these systems get older and the mineral surfaces get more crystalline, then there's less opportunity for that inositol phosphate to be stabilized. So what happens is it's broken down, it's mineralized, it's broken down by enzymes. So that's what we think is the explanation, is at least part of the explanation for why inositol phosphate, for example, decreases so dramatically towards the end of it, towards the older part of the system. The DNA part of it is a little. Now, if we look at the diesters, then we get exactly the opposite to what we expected. We expected these diesters to be very small proportion and probably to decrease with time as the system gets older. What we found was exactly the opposite, that the diester phosphorus, certainly a DNA-associated diester phosphorus, increases quite steadily with time. Now that's a little bit of a puzzle. And so, to cut a long story short, this is a very complicated diagram, but it took seven of us 5 years to actually, about 4 years, to pull the data together. Because what we did was we took the Franz Josef chronosequence and we basically collected all the data that we could find on plant and soil and this is actually to 1 meter depth. Yeah. So these are actually in grams per square meter, or you can scale it up to kilograms per hectare. And what we did was we split. It looks a bit complicated, but it's actually quite simple when you end up, because basically we identified 6 pools of phosphorus. This is the inorganic pools. These 3 here: the grey, the light blue, and the dark blue. The grey, and I'll use this 1,000 years to illustrate this. The grey is actually apatite phosphorus, calcium primary mineral phosphorus, that's associated with stones. In other words, material that's greater than 2 millimeters, right? So this is not soil. The definition of soil in strict terms is something that's less than 2 millimeters in size. So this is a calcium phosphate that's in stones. The light blue is calcium phosphate that's in soil, so it's in less than 2 millimeters, and the dark blue is phosphorus associated with iron, inorganic phosphorus associated with iron and aluminium. So you've got primary mineral calcium, apatite phosphorus associated with stones, basically. You've got the soil calcium phosphate, or the apatite, and then you've got the iron and aluminium. We over to the biological side of things. The green square is actually phosphorus in the plant biomass. The brown square here is actually phosphorus in organic phosphorus in the soil. And importantly, the little red triangle there is phosphorus in the microbial biomass. So we have these 6 pools, and we have them over 4 time periods. So we've got initially 5 years, 1,000 years, 12,000 years, and 120,000 years. So you can see the progression here in terms of the size of the pools is relative. It is relative to the size of those squares and the triangle in the case of microbial biomass is relative to its size. So you can see at the beginning we have most of the phosphorus is actually contained either as calcium phosphate in the rocks or in the soil. There's very little biomass. When we get to 1,000 years, we've lost phosphorus from the system, but you'll see that the biomass pools are developing. We've got phosphorus in the biomass. We've got a large microbial biomass pool and an even larger organic phosphorus pool. But if we move forward to 12,000 years, we're starting to move into that retrogressive. You'll notice that the rock is actually broken down. So these systems, the rocks actually break down over time. So you can see that most of the phosphorus is now inorganic phosphorus dominated by iron and aluminium, and that would be stable iron and aluminium phosphate. The biomass is there. It's probably at its somewhere near its, probably past its highest point. Organic phosphorus is about the same between 1,000 and 12,000 years, as in the microbial biomass is about the same. But if we go forward to 120,000 years, we see that the biomass has collapsed. We see that there's very few rocks left in the system. Organic phosphorus has declined, and so has microbial biomass. But the important point is that as a proportion of the total phosphorus in this system, the microbial biomass increases dramatically when you go from 12,000 to 120,000 years. So what this tells us is that when these systems move and get very old, as these systems get older, the microbial biomass becomes more and more and more important. And that accounts for the fact that we could see this increase in specifically in DNA associated organic phosphorus with time. So it illustrates the importance of in very low phosphorus and phosphorus availability environments, the importance of the microbial biomass and maintaining. But let's go to agricultural systems to finish this off. It's very difficult actually speaking to a screen. I'm used to speaking to people. But let's talk about agro-ecosystems and managed ecosystems just briefly. And I mean, standard agronomy, and this is for New Zealand pasture systems. What we're trying to do is we're trying to achieve levels of available phosphorus in the soil, in this case, Olsen P, soil test P, sufficient to obtain about somewhere between 75 and 95%. So the basic idea of fertilizers is that you put in an initial fertilizer to increase your fertility to somewhere that to achieve somewhere between 75 and 90% of maximum relative yield, and then you're going to put phosphorus in every year to maintain it, to account for losses and transfers, etc. And so when we do this in New Zealand systems, the more intensive the system, these are the annual maintenance fertilizer requirements for different systems. And we go all the way from plantation forestry, which doesn't require any phosphorus. If we look at pastoral systems, if you look at very extensive hill country systems, they only require about 20 kilos of phosphorus to be maintained. We go into flatland, we can increase the amount of production, so we need more phosphorus. When we look at dairy farm systems, these are very much more high production systems. We need 50 kilos. And then you move into cropping systems, into high crop systems like potatoes, 75. And then if we go to vegetable production, where you're doing two, three crops a year, you're looking at 150 kilos. So these are the amounts that are required to maintain these systems. And one of the things about maintenance, about this maintenance of fertility, and this is the same all over the world, is that the systems are quite inefficient. And if this is an example from Australia, and it would be similar in New Zealand, if we look at the phosphorus use efficiency, in other words, of that phosphorus that we're putting in for maintenance, how much of it is actually utilized in the system, then we can see that cropping systems are not bad, 48%, dairy 29%, but we look at grazing, extensive grazing systems, we're down to 10 to 20%. And what that means is that we're getting surplus, and we're getting surpluses that accumulate. And if you look across those systems, you can see that somewhere between, the average is around about 10 up to 20 kilograms of phosphorus is accumulating in these systems per hectare per year. And part of that is just an inefficiency, it's an inherent inefficiency in terms of plants being able to access that phosphorus. But a major factor is fixation or retention of phosphorus. In other words, absorption of inorganic phosphorus onto mineral surfaces mainly. So there's a phosphorus use efficiency is relatively, it can be very low for productive systems. And if you look at another example, this is a dairy farm in England. I'm not going to go into detail, but essentially you've got 58 kilos coming in to maintain the system. You've got 33 kilos going out, which means that you're accumulating 25 kilograms. 25 kilograms per hectare per year is accumulating in those soils. And that's in a highly productive system. But if you look at the system, never mind that, just leave that, doesn't matter, that was another example. But we can actually look at this globally as well. And this is data from 1965 to 2007. And the blue column is P inputs, and the green column is P outputs. So just focus on the figure at the end here, which is globally. So globally, we've got basically half the phosphorus that we're putting in. We're only recovering half of what we're putting in. So over that time period, we've accumulated around about, on average globally, and it's going to vary, of course, widely around the globe, about 250 kilos of phosphorus per hectare. And this is collectively known as legacy phosphorus. And there's a great deal of interest in terms of trying to improve efficiency and mobilize some of that legacy phosphorus. If I come back to agronomy again, this is a very similar graph to put on before. Basically, this is Olsen P, or available P in the soil. This is the agronomy. So you get up, and that's that figure there is about your 90% relative yield figure. That's what you're trying to attain. This red number here is actually phosphorus in drainage water, in drainage. Okay, this is dissolved reactive phosphorus in drainage water. And what happens is that as we put more and more phosphorus into the system, if you increase the phosphorus inputs into the system, then what happens is you get more and more accumulation of phosphorus. And so as you accumulate more phosphorus in the soil, then the amount of phosphorus that actually is released in drainage increases. So this accumulation of phosphorus that happens as a consequence of this maintenance fertilizer, even if you're trying to maintain it there, you're still going to accumulate phosphorus in the soil, and you're going to get a potential increase in drainage. And the reason why that's important is because if we get more phosphorus and nitrogen into waterways, we get accelerated eutrophication with all its incumbent problems with weeds, with algal blooms, and all sorts of problems. So there's a lot of incentive to actually improve phosphorus use efficiency and reduce losses, drainage losses in systems. So if we've got phosphorus levels in a lot of soil in soils where we've got levels of phosphorus that are up in here, accumulated phosphorus, then we want to get back and mobilize some of that legacy phosphorus if we can. The point about it is, so this is what we're talking about in terms of strategies to mobilize lower legacy phosphorus. The first part is relatively easy. If you've got a system where you've got very high levels of phosphorus, then to get the levels down to to reduce the levels, you simply reduce or eliminate, not necessarily eliminate, but you reduce the amount of phosphorus that's going in, and the system will, the levels of phosphorus will fall. But if you want to lower that further, for example, if you set a critical level below that, that is trying to improve the P use efficiency, then getting this bit out is really difficult. This is where the challenge really is, because mobilizing phosphorus down from very high levels to medium levels is relatively straightforward and just takes a bit of time, but getting it further down requires a bit more effort. And we've got one trial here that I set up way back in 1994, and it wasn't set up to look at this, but it was set up for many reasons. And it's basically a mixed pasture system that was set up, no animals, and there's a number of treatments, but the two main treatments is basically the grass is mown or the biomass is removed. In other words, it's taken off site, or it's mown with the biomass retained. So basically you've got an exploitive system and you've got a return system. This is almost like a graze system. This is almost like a crop system. So after 20 odd years, so we're looking at approximately 20 to 22 years here. If we look at the red is the retained biomass retained and the blue is the biomass removed, we see that we've drawn down the total phosphorus, and inorganic phosphorus has gone down. The plant available inorganic phosphorus has been dramatically and significantly reduced. So basically what we've done in terms of inorganic phosphorus, if I go back to here, is that we've achieved strategy A. However, with regard to mobilizing, for example, organic phosphorus, nothing happens. We get no mobilization, despite the fact that we're stripping phosphorus out of the system. There's been no change in organic phosphorus after 20 years. And just to summarize that, if we look at the inorganic phosphorus, this is after 20 years, if we look at the inorganic phosphorus, the different pools, the very soluble pool going into the lower, less available pools, we've actually extracted about 30 to 60% of the inorganic phosphorus. We've mobilized 30 to 60% of that phosphorus. We've mobilized some organic phosphorus, but not very much. And when we look at the majority of the organic phosphorus in the soil, even though the available phosphorus levels are very, very low, we have not yet seen mobilization of organic phosphorus. And so that's a real challenge. I'll come back to that in a minute. So how do we mobilize? How do we, if we want to mobilize some of that organic phosphorus in particular? And there are a number of studies that have shown that mobilization of organic phosphorus is really quite a challenge. And there are a number of different ways that possibly go do this. I haven't got time to go through all the options of increasing phosphorus use efficiency in agro ecosystems. We wrote a paper on advances in agronomy a few years ago on this. But I just want to focus on a few examples in terms of looking at biological processes around enzymes and particularly organic anions as well. And one of the theories is what we call the Unbutton model, which was developed by Murray and Clarholm in 2015, which basically tried to give us a model or a system that we could work on, whereby plants, plant roots, fungi, and bacteria. The theory here is that it's actually a mobilization of organic phosphorus and nitrogen. So here we've got nitrogen and phosphorus in organic complexes. These organic complexes are held together by mineral cations like iron, aluminium, and calcium. And the release of inorganic nitrogen and phosphorus in these systems is actually a two-stage process. One is the release or the exudation of low molecular weight organic anions like oxalates and citrate, etc. And these actually sequester the calcium, iron, and aluminium. And that basically frees up or unravels, if you like, exposes the phosphorus that's associated with organic matter and nitrogen. And then we have the enzymes that are produced by these organisms, and that is what breaks down and releases the nitrogen and phosphorus. So it's a two-stage process. And so there's a very important role for organic anions or they are organic anions. People refer to them as organic acids. They're not acids at most soil pHs. They're actually organic anions. And so it's a two-stage process. So if you want to mobilize organic phosphorus, legacy organic phosphorus, I'll just skip that one. Then one of the things you have to do is to look at changing the plant or changing the biology or changing the mycorrhizae. And I'll just give you one example of this, which is another trial that we set up in the late 1990s, where we were looking at established grassland with high legacy phosphorus, high levels of legacy phosphorus. And what we did was we put different species of tree in there, and radiata pine and a eucalypt with different mycorrhizal associations. And what we found was after 20 years that just changing the plant, we mobilized, as you can see, a large proportion of the available phosphorus. But what was really important was we actually were able to mobilize large amounts of organic phosphorus relative to the pasture. So this is just an example of how changing plants, changing mycorrhizae, changing their associated microflora can be used to mobilize organic phosphorus. Another option looking at cropping systems is to use green manures. And I think my personal opinion is that this is where we have the most potential to actually make a difference in cropping systems. So this is just an example of some what we did recently using different green manure plants and looking at them in legacy phosphorus and mobilization of legacy phosphorus, short-term mobilization. You can see they've got different abilities. You can see that lupins are by far they mobilize the most phosphorus compared to peas and buckwheat. But what's also interesting is that the lupins mainly mobilize inorganic phosphorus, whereas, for example, pigeon pea and chickpea actually, well, particularly pigeon pea, mobilizes organic phosphorus. So that suggests that there's different mechanisms going on here, and possibly going back to that model that we talked about. The other thing I just want to mention briefly was some work we did quite a long time ago, looking at these green manures. And this was using a lupin green manure and maize and doing simulating multiple crop rotations. And one of the things about green manures is you grow them. Oh, I should have explained, just in case. A green manure is basically a crop that you grow between crops. And you don't take the crop, you grow the crop, you don't let it mature. It's a very immature crop, and you basically plow it in. You return it to the soil. So you take the phosphorus up and the nitrogen and whatever, and you return it, and the carbon, and you return it. And what we found with this system was that using some quite complicated methodology, we found that after 2 cycles of green manure, compared to fallow, just leaving it the ground without plants, this is a measure of the mineralization rate of organic phosphorus, and we found that it had gone up almost 5-fold. It was 5-fold higher in green manure, as a result of green manure. So that there's a potential there for green manure to enhance biological cycling of phosphorus and so in cropping soils as well. And if we can tie that in with mobilization of legacy phosphorus, then I think we have some potential very useful techniques. So just moving on to some conclusions. Basically, organic and biomass phosphorus account for over half of the total phosphorus in most soils. The biological processes play a critical role in determining bioavailability. Optimizing biological phosphorus cycling, this is my opinion, is required to increase legacy phosphorus mobilization, enhance phosphorus use efficiency in agri ecosystems. I think that's crucial. However, there's a lot we don't know. The turnover, the flux of phosphorus between various biotic pools, interaction, and the role of extracellular enzymes, and I should have mentioned organic anions, is really quite poorly understood as yet. And again, this is my opinion, improved phosphorus use efficiency. I don't think there's any magic bullets or any magic solutions. It'll be driven by the actions of plants and their associated microflora, whether that's mycorrhizae or whatever. So thank you for enduring that. I went on a bit longer than I expected. Thank you.
Well, thank you very much. Thank you very much for enduring not sleeping, and you're there up very late. And thank you very much for this excellent presentation. I'm sure that many of our viewers are appreciating it, as it shows from the chat box. Now we'll go, maybe we'll pick up a few questions before letting you go. And I guess there was a question about some reference to the soil formation that you showed, and somebody has missed that. Could you show the slide with the reference so that the people can take note?
With the reference? All right, okay. Just a minute. Hang on a sec.
Soil formation, yes.
Yeah, wait. If they contact me, I can send them references, not a problem.
Okay, very good. So the viewers,
I think you're referring to this one. I think it's referring to, well, where is it? That one. I think it's referring to that one. Yeah. Okay. That's a very famous paper published in Geoderma. Okay.
Yeah. Okay. I guess the people who are interested, the viewers who are interested, will contact you for references.
No problem. You're welcome.
Yeah. And here is another question, I guess about the same. They want to know the source.
The source of this of the information. Yeah. Well, again, email me and I can send you any questions you've got. I can send you the papers or the links or the
okay,
references.
very good.
No problem. It's all public. Public. Yeah.
Okay, very good. Here's another question by, I guess there was by one of our scholars at UM6P. And he says, thank you, Professor, for presentation. And his question is, how microbiology with mycorrhizae can contribute to solubilization availability of phosphorus?
Yep, I think it's got a crucial role, and I think we need to identify and quantify the role of different mycorrhizae and in mobilization. I think they're crucial, and as they illustrate the figure I gave there of the different tree species mobilizing organic phosphorus is an example of that. But I think in cropping systems it's a little bit more difficult because you're dealing mainly with AM mycorrhiza. But I still think it's possible, and I think it's a question of identifying the mycorrhiza that are best suited to mobilization, and then possibly trying to introduce them through inoculation of seeds, etc. So I think that there is potential there, but it's not easy. I mean, the whole mycorrhizal association, even in cropping soils, is very, very complex. But it's a real challenge. But I mean, so some of it, and in any of these systems, you know, the mycorrhiza is so intimately associated with the plant I'm beginning to think that we shouldn't be talking about necessarily, but we talk about plant mycorrhiza associations. They're so dependent on each other that, that they're absolutely crucial. So I think it comes down to plants as well as mycorrhiza, and how much we can manipulate them. There's definite avenues there for progress. Yeah, and progress is being made. Yeah, definitely. Yeah.
Okay. There is another question by one of our PhD students, Mr. Masamba Diop. And he says, many thanks. And his question is, what are the best options to build up organic phosphorus in soil in a long term? And what manage to allow bioavailability of this capital to crops?
Okay. I think it's all about carbon. It's all about plants. The more biomass you have in the system, and the more you can encourage the recycling and avoid things like fallows, etc., and try and keep as much plant material and encourage. That's why I think green manures are great, and cover crops are potentially very, very valuable in terms of encouraging biological cycling in the soil, and that in turn may or may not have major improvement in terms of P availability and utilization of phosphorus. We really need a lot more data to know whether the use of cover crops or the use of green manures is to quantify their impact. We know in the lab and short-term studies that they do make a difference, but the field work, there is work going on and there is evidence starting to come through. But it's basically, if we can encourage as much biological activity as possible, then I believe that there's one way of improving or potentially improving P use efficiency and overall phosphorus cycling. Yeah.
Thank you.
No worries.
This question is by Ainaram. I guess he must be from Madagascar, one of our friends from Madagascar.
Sure.
He says, thanks for the presentation. It is a very interesting topic. Is the presentation in PPT or PDF available for sharing?
No problem.
Yeah, I
No
guess.
I can send it to you, Mohammed, if you want.
Yes, please.
And then anybody can contact you. I'll send you the PDF copy tomorrow. Yep.
Okay, great. So we'll have it available for our students
if anybody wants it, if anybody wants it, they can contact you. Yep.
Yeah, our digital expert, Mr. Usama, will do the job forwarding your presentation to anyone who would ask for it.
No problem.
Yeah. Okay, very good, super. So let's see if there are any more questions. Yes, this question by Mr. Sohib Abdul Rahman. He says, is there side effects on plant root if I add overdose of organic phosphorus to soil? And what can I do to solve this problem if it is found? Yeah.
I've never heard of an overdose of organic phosphorus. I've never actually heard that before. But I think what you mean is if you oversupply organic, if you put too much organic phosphorus in there, you get accumulation of organic phosphorus. I think the only thing to do is to try and avoid that situation or reduce the inputs of organic phosphorus, or use systems that will mineralize organic phosphorus, looking like cover crops, etc. You know, the other thing about cover crops, it's not necessarily about. It's about improving or enhancing the overall turnover of phosphorus in the system. So, and it all links back to carbon. It's all about carbon availability as well. So I'm not aware of that being a serious problem in terms of overdosing with organic phosphorus. And I think I know what you mean, but I don't think it's a big problem.
Okay, well, there are quite some questions, and this one is from Jean Damascene Tuizia. And he says, Professor, could you comment on the variation of phosphorus level between parentheses, production, consumption, and under agriculture of conservation compared to ordinary farming system? With thanks.
Yeah, I'm assuming that conservation means lower input, lower production systems. I mean, there's also a lot of talk. There's also a lot of talk about regenerative agriculture systems as well, in terms of trying to use a slightly less intensive approach to production. And I would imagine that the phosphorus level, I mean, the thing about phosphorus levels is, and I think we have to remember this, is that for many, many years, phosphorus was very cheap in agriculture. It was incredibly, I mean, the price of phosphorus even into the early 2000s was, in real terms, actually cheaper than it was in the late 1940s. And to be honest, I think that encouraged a little bit of overindulgence in many areas of the world, particularly in the West, in terms of in Europe and North America and Australasia, in terms of the use of phosphorus. So when we look at a conservation approach, again, it comes back to this. A lot of the phosphorus that's been added to soil in the past is still there. So it's this legacy phosphorus. So in terms of a conservation approach, a lower input, possibly using different combination of plants and rotations, you can mobilize and you can utilize phosphorus more efficiently. So I think I think the idea is that if you've got accumulated phosphorus, or if you've got very low levels of phosphorus, this is the other thing about mobilization, and it's something that people like myself are going to be very careful about, because I mean, I work in systems where in agricultural systems where phosphorus is not really limited, and we've got problems with too much phosphorus. But we realize that there are large parts of the world, and including large parts of Africa, where that is not an issue. The issue is very low levels of phosphorus. And this is where it comes into using these biological systems to optimize phosphorus use efficiency, where you've got very little phosphorus, or you've got very limited availability of phosphorus or access to phosphorus. You have to use what you get and what's in the soil very, very efficiently. And I think what you mean by conservation is a much more biological and a bit more using different systems to maintain production on these systems. So I think that there are ways that you can manage phosphorus better in these systems. Yeah.
Okay. There is another question from Mr. Rashid Lanaya from Morocco.
It's a very good question. I mean, I don't really know. I mean, the thing about it is that the mycorrhiza depend on the plant. So it's and I mean, the mycorrhiza drive the plant drive the mycorrhiza. It's a symbiotic relationship. There is a lot of evidence to show that it's actually the mycorrhiza that's driving a lot of this. So it's the plant, because the plant supplies the carbon. So it comes back to basic soil fertility. If the soil fertility is right, the plant will grow, the mycorrhiza will thrive, and you'll get you'll optimize your use of things like phosphorus and nitrogen. Yeah.
Very good. And I'd like to close with this. Our friend from Madagascar is thanking you very much for this excellent presentation, and we're happy that we have been joined by people from different countries. And thank you very much for your time again, and for your bravership to stay late at night to
No
share
problem.
to share with us your knowledge.
No problem.
Please convey our heartfelt greetings from UM6P and Morocco to people of Lincoln University and the whole people of New Zealand.
Okay. We'll do it.
We are, despite the distance and despite the distance, it's amazing how much the new communication technology can bring the people together, not only overseas, but we'll be looking forward to having you here, Morocco. XP. Thank you for hosting our students. Thank you again for all the time you have devoted for their education and for sharing with us your knowledge and science.
Okay, it's been a pleasure, and I just wanted to say that a number of the aspects that I talked about today, because the 4 PhD students we have here at the moment who are just finishing their PhDs at the moment, they have all worked on various aspects, or 3 of them anyway, related to what I've been talking about. So they've made a significant contribution to this research as well. So it's good.
Super.
Okay, well, good morning or good afternoon or good night or whatever. Yeah.
For us, it's good afternoon.
Okay. Right.
And for you, it must be good morning, and we must say good night because you must get some sleep.
Okay. Right.
Okay.
Good night. Bye.
Good night. Good friend. See you some other time.